welcome to the ELK Molecular Biology Lab. Today, we will be working together on a technique crucial to genetic testing and medical diagnosis: real-time fluorescence quantitative PCR. PCR is a method that uses fluorescent chemicals to measure the total amount of product after each polymerase chain reaction PCR cycle. It uses internal or external references to quantify specific DNA sequences in the sample. Combining PCR technology with real-time fluorescence detection, it monitors DNA amplification in real time during the PCR reaction, enabling quantitative analysis of target DNA. Before the experiment, we need to prepare the following instruments and consumables. Next are the required reagents. The purpose of this experiment is to detect the changes in the relative expression of the target gene S in clinical samples numbered 1 to 33, using GAPDH as the internal reference gene. So let's get started. First, let's set up the qPCR system. Note that we performed 4 replicates for each gene. To reduce experimental error, we first mixed all reagents except the template. Because pipetting can cause loss, we needed to prepare a larger amount of each component. For this experiment, we prepared 2 systems. Prepare 2 1.5 milliliter centrifuge tubes, label the tube caps with GAPDH and S genes, respectively. Prepare the qPCR systems for GAPDH and S genes according to the following system. Use a pipette to pipette the following reagents into the GAPDH centrifuge tube. 700 microliter NT-Link SYBR Green PCR Super Mix. 140 microliter GAPDH 2.5 micrometer primer working solution. 252 microliter DD water and 28 microliter ROX. The S gene operates in the same way. After adding liquid, place the tube into a vortex machine for vortexing, centrifuge, and finally place it in an icebox. Use a pipette to transfer 8 microliter of each mix from the GAPDH and S gene qPCR system tubes to a 384 well plate, with 4 replicates for each sample. Transfer 2 microliter of the diluted cDNA to the corresponding wells. During the experiment, please note that after each addition of cDNA template, the sterile pipette tip must be replaced to avoid cross-contamination of samples and ensure the accuracy of the test results. Use the same step to spot the S gene. After spotting, we're ready to seal the plate. It's important to note that to prevent fingerprints from getting on the sealing membrane and affecting experimental results, disposable non-slip polyethylene CPE gloves should be worn during subsequent operations. Seal the completed 384 well plate with a sealing cover. Press the edges tightly and place the sealed 384 well plate in a microplate mini centrifuge for centrifugation. After centrifugation, place the 384 well plate into the Life Technologies QuantStudio 6 Flex Fluorescence Quantitative PCR instrument for on-machine detection. The setup procedure is as follows. Run the machine. When the fluorescence signal in the reaction tube reaches the set threshold, the recorded PCR cycle number is the CT value. After the reaction is completed, the instrument will automatically generate data such as CT value and melting curve. Through these experimental data, the relative expression of the target gene can be accurately analyzed, and the relative expression level of the target gene in the test sample can be determined. The key to a successful experiment lies in standardized operation and rigorous analysis. I hope everyone can master this important molecular biology technique.